All right, so we are going to talk now about cell membranes, and you have a couple of different cell membrane structure. So I put all of those kind of together. The first is I can list the four main parts of a cell membrane, and so that is going to be your phospholipid bilayer, that's the bulk of the cell membrane, proteins, cholesterol, and carbohydrate chains. So those are your four parts, and I can identify those in a picture. So if you were given a picture, y'all know my art skills aren't great. You also should be able to draw a picture, although you probably won't have to do that other than for your portfolio. So there's an example of a cell membrane. And so what we see in here is we see our phospholipids. So that's our phospholipids. We see a protein. Now I made an integral or transmembrane protein, one that goes all the way across the membrane. You could also have a protein that's just kind of in half the membrane. That's called a peripheral protein. Think about your peripheral vision. You also have carbohydrate chains, and those can be coming off of a lipid or they can be coming off of a protein. Okay, and so those are our carbohydrate chains. And if they're coming off, do that. If they're coming off of the lipid, they are called a glycolipid. Glyco just means sugar, so it's a sugar attached to a lipid. And if they're coming off of a protein, they're called a glycoprotein. You may not need to know those words, but I'm going to share them with you just in case you see them somewhere. You'll be like, oh, I know what that is. All right, so I can identify the four main components of a cell membrane in a picture. So that's three of them: the phospholipids, the protein, the carb chains, and then the other one is the cholesterol. Now, I'm not good at drawing those little six-sided or five-sided molecules like for chemistry, but cholesterol is a four-ring structure. It's a lipid, but instead of being like a carbon backbone, it's a ring, and it's four rings hooked together, so it's a steroid. And so, let me blow this up a little bit, that helps. All right, and so you're going to see that kind of in between your fatty acid tails, because it is going to help provide stability and keep those tails from tangling up. All right, so those are your four parts. You can identify them with a picture. I can identify the hydrophobic parts of a cell membrane, of a phospholipid. This is your polar head. It is a phosphate group. And anything that is polar, because water is also polar, is going to be hydrophilic or water loving. Anything that is non-polar, because we know that polar and non-polar things do not like to hang out. Water's polar, so if this is non-polar, it is not going to like water. And so it is hydrophobic, okay? Anytime you have both of those conditions, hydrophilic and hydrophobic, on the same molecule, we call that amphipathic, okay? Amphi means both, path means condition. So we have both conditions on the same molecule. All right, so I can identify the hydrophobic portions, I can identify the hydrophilic portions, I can describe why it's called amphipathic, and then I can explain why the current model of the cell membrane is called the fluid mosaic model. All right, so this model that we just drew up here is known as the fluid mosaic model. And when you think of fluid in this kind of situation, we're not talking about a liquid, we're talking about fluid as in movement. So when you say something's fluid kind of as an adjective, that means it can move. It's not rigid, it's not hard or stationary, it's movable. And that's how the cell membrane is. It undulates. I don't know if I can get that. Not really. With the camera, it's kind of hard. But if you picture like wind blowing through waves of grain, it kind of creates this wave-like pattern, and that's going to be an undulation. And so you see that in the cell membrane. You also see phospholipids switching places. So they will move horizontally. So this guy would switch place with that guy. They just kind of swap. You can also see them moving vertically. So this one can swap. So they'll actually literally flip that way. And so there's a lot of movement in the membrane, and that allows things to be moved that need to be moved. And so that is the fluid part of fluid mosaic. The mosaic part is a mosaic, if you think about that as an art term, it is where you use little different pieces of something, paper, tile, whatever, and you use those different pieces to create your art, to create different images. Well, you're not creating images in your cell membrane, but these proteins are scattered throughout. So when you look at a cell membrane, you see all of these little phospholipids, and then you'll see this protein there, okay? If I had drawn it bigger, what you would see is you would see them scattered all over the cell membrane in this kind of mosaic. It's not a pattern, so mosaics tend to be, when we use them in art, we use them to make a pattern or an image. That's not what it means here, but it is scattered like a mosaic. I am going to pull one other learning target that's a little further down in the same group of learning targets, but it's just a little further down. I can discuss how organisms can alter their membrane makeup in order to respond to their environment. All right, normal membranes, and I'm not going to draw all the other players right now. I'm just going to focus on the phospholipids. Normal membranes have phospholipids that have saturated fatty acid tails, okay? And so saturated, if you will remember, saturated means you have all single bonds. So all of these carbon chains, which is what these fatty acids are, they're just a bunch of carbons linked together, they are going to be straight, and they're going to be able to be tightly packed. So they can, because their little tails are straight, they can pack really tightly together, which is great. However, it is going to cause some rigidity to the membrane. It's going to decrease that fluidity or that ability to move. When it gets cold, cold temperatures increase rigidity. So it's going to make it even more rigid, even more unmovable. And so if it were to be bumped or moved or something, it could fracture. And then when the cell thawed, then all the contents would spill out and the cell would be dead. So that's not what it wants. So what some species can do, not all, but winter wheat is an example. Wheat as in what you make bread from. Winter wheat can actually, when the temperature starts getting cooler, it will start switching out. Oh, I shouldn't have drawn those. Ignore that. It will start switching out some of these tails for unsaturated. So not all of them. There's still some saturated ones in there that are straight, but it's starting to move some of these or pop out some of those unsaturated or saturated and replacing them with unsaturated. So when you do that, now what you're seeing, and I forgot to draw my little ones down here. Let's say those are all still saturated. When you do that, you're going to start to see those bends in the molecule. So they're going to be more loosely packed. And that's going to give some flexibility to the membrane. So it's going to be more flexible. And so that's what winter wheat and some other species do. They switch out the saturated for unsaturated, and that allows, so when that cold temperature makes their cell membrane more rigid, they've already made it more flexible first. And so when it becomes a little more rigid, it doesn't hurt the cell like it would if it was all saturated. So that is how organisms can alter their membrane makeup in order to respond to the environment. And that is all about cell structure. I'll have another, or cell membrane structure. I'll have another video in a second on function.